If a box and whisker plot showed up on your AP Bio exam, would you even know what the box means? Hi, I'm Ms. Parrott, and in this video, we're going to be looking at box and whisker plots for AP Biology. Box and whisker plots are different than bar or line graphs because they're all about showing data distribution. This is an example of one. The goal of this video is to help you learn how to interpret the data given in this kind of graph efficiently and accurately. We use box and whisker plots to compare the median of sets of data, spread, and overlap for different groups, or categorical data. First, let's look at the parts of a box plot. You have the box, and you have the whiskers. So these are not error bars, they're whiskers, and they have a different meaning. Then you have this line in the middle. The line in the middle is the median, the number that is in the center of the data if you lined it up. This is not the same as the mean, it's the median. The box shows the middle 50% of the data, so it's the range of the middle half of the data. We call the line below the median line, the bottom part of the box, Q1 or quartile 1. This means that 25% of the data are below this number. And then the top is Q3 or quartile 3, and that's kind of like your 75% mark. So 75% of the data are below this mark. The top whisker is the max value, and the bottom whisker ends at the minimum value in your range of data. So that's what these are, but what does it mean in a graph? Great question. So first, we don't want to get whisker and error bars mixed up because they do look a lot alike, but they have very different meanings. Again, whiskers are showing us the spread of the data, the minimum and the maximum, whereas error bars are showing us the variation around a mean, right? So on a bar graph, you have like the top of your bar is the mean, and then your I bar or your error bar shows you the variation around that mean. Usually you have to use standard deviation to determine the standard error of the mean, and it'll be plus or minus 1 or 2 standard error of the mean. But this is different. These are actual, like your top and bottom numerical value. So what's important about this is that when you're writing your conclusions, you don't want to say, when you're looking at a box and whisker plot, the error bars overlap, because they aren't error bars. They're whiskers. Better wording would be something like the ranges overlap in group 1 and group 2, or the boxes overlap, or even the medians differ, but the ranges overlap. So be sure when you're describing what's happening, you're using the correct vocabulary. When you're interpreting a box and whisker plot, and we're going to do an example in just a second, you want to ask yourself 4 questions. One, because you're comparing, this is always for categorical. You use these on categorical. So you're comparing different groups of data. So which group has the highest median or the lowest median? Which group has the greatest spread? Do the ranges or the boxes of the groups overlap? And what claim is actually supported by the data? So let's try that with this. So this is a leaf decay example. This is pulled from the AP Bio course exam and description from 2025-2026. And it's looking at 3 different tree species, ash, sycamore, and beech. And then the people collected a bag and saw which leaves decayed the quickest or decayed over time. So it checked percent of decay per bag. Right, we have our 3 species, and then here are the data. Again, the means and the ranges, the maxes and the minimums for each species. So which had the highest median? For median, we look at that middle bar. So the highest median would be ash at a little over 50%. The lowest median was beech, down here at 20% of decay per bag. Which had the greatest spread? So which one? That would definitely be ash. It had a greater spread than either of the others. Do any of the ranges overlap? So in this case, ash and sycamore, even though the median of sycamore is lower than ash, their ranges definitely overlap. So what claim could we possibly support? So if the question was which one decayed, like the most quickly or the slowest, we could say that beech leaves decay more slowly. That would be a claim that's supported by the data. It's slower because less has decayed, and they're all given the same amount of time. And so that's our claim, and now we want to justify our claim with evidence from the graph. So we can say the median and the range are lower in beech leaves than either sycamore or ash. If you wanted to follow up, you could say, while the median for sycamore is lower than ash, the ranges overlap, so it's difficult to determine if ash in fact decays quickest. More experiments would need to be done. Simple. Common mistakes made when dealing with box and whisker plots. Just remembering median and mean are not the same. Median, right, we use that when we're trying to get rid of outliers or not let outliers sway the story of our data, which can happen when we use the mean. Whiskers are not the same as error bars. Error bars show variation around a mean. Whiskers show the maximum and the minimum in your data set. Overlap does matter. So whether if your whiskers just overlap, then that means you just have a little overlap in data. But if your boxes overlap, that means there's a considerable overlap in the data from two groups. So what overlaps and how much matters. Also, you don't want to claim a significant difference without a statistical test like a chi-square analysis. So even on that last board where we were looking at ash and sycamore, yes, the medians are different, but since there's so much overlap, you would want to do a chi-square test on that data to make sure to see if the difference is statistically significant or not. It is okay if all this box and whisker stuff is new or you're not familiar with it yet. Your job is to keep practicing with box and whisker plots, and that's what's going to lead you from, after this video, you know about them, to you can make them or you can understand them, to you can do it quickly and easily in an FRQ on an AP Bio exam. That's what fluency is. If you found this video helpful, please do give it a like, subscribe to my channel, Miss Parrot Science, and please do leave a comment, a question, or an emoji down below. If you want more graphing practice, please check out my Teachers Pay Teacher resources at Know and Grow with Gnomes on TPT. I just put out, so for all you teachers, very excited, I just put out a AP Bio complete math bundle that includes all of my formula and equation sheet resources in one place. So you can get your whole year's resources all at once in one place before school starts. Keep learning, keep growing, and we'll see you next time.